Hello guys, welcome back to my channel. I hope you all are doing well. Today we are going to talk about microbial growth phase, kinetics and factor affecting growth. So without any further ado, let's begin with the PPT. Understanding the dynamics of microbial population and environmental condition that influence their growth patterns. So first of all, what is a microbial growth phase? What is the meaning of this word? What does it mean, and what is the importance of studying this or even knowing about this thing? So guys, microbial growth phase refers to the distinct stage that a population of microorganism like bacteria goes through when when cultured in a closed medium such as test tube or fermenter. So the need of microbial growth phase, or the need to study the microbial growth phase, is to know how the population of microorganism like bacteria grows in a closed medium. Closed medium is what is happening with the microorganism when that microorganism is inside a test tube, or when the microorganism is inside a fermenter. How is it growing? What are the phases a certain microorganism is going through? So this is the reason why we study microbial growth phase. So with limited nutrients and no removal of waste products, so when the microorganism is going through the microbial growth phase, they usually have limited nutrients and no removal of waste products. These phases are usually illustrated in a bacterial growth curve, which includes four main phases. So guys, there are four main microbial growth phase which I'll be talking about in this PPT. Also, I will be showing you the graph which demonstrate a microbial growth phase. So moving on to the next PPT. Understanding microbial growth from single cell to population. Microbial growth refers to the increase in cell number rather than the cell size. Unlike multicellular organism, microbes reproduce through binary fission, creating exponential population increases under optimal condition. So each generation doubles the population, making rates critical for application ranging from biotechnology to food safety. Understanding these patterns enables precise control, industry process, and effective antimicrobial strategies. As I said, microbial growth phase refers to the distinct stages that a population of organism like bacteria goes through when cultured in a closed medium, right? And it has four phases. In simpler words, microbial growth phases are the different stages that microorganisms go through when growing in a closed environment. These include a period of adjustment, fast growth, or no growth, and eventually cell death. So there are four phases of microbial growth curve. As I said, there's four phases I will be talking about. The first phase is the lag phase, then comes the log phase, which is also known as the exponential phase, then comes the stationary phase, then the last phase is the death phase. So there is lag phase, then there is log phase, which is also known as the exponential phase, then there is stationary phase, then there is death phase. Each phase, lag phase, so there is different process. So according to the lag phase, adaptation period with no net increase in cell number. So there's four phases: lag phase, exponential phase, stationary phase, and death phase. And among these four phases, everything different is happening with them. There's something else happening in the log phase, there's something else happening at the exponential phase, there's something else happening at the stationary phase, and there is something else happening at the death phase. So let's see what's happening at the lag phase. So usually the lag phase, adaptation period with no net increase in cell number. So what usually happens at lag phase is the microbes adjust to the new environment, no growth there. So this is the initial phase. The lag phase is the initial phase where microbes start adjusting to the new environment and there is no growth rate yet. Then comes the exponential phase. The exponential phase is also known as the log phase, where the rapid cell division at maximum growth rate. So over here the microbes grow and divide rapidly. So over here the microbes start growing and dividing rapidly. Then comes the stationary phase. At the stationary phase, what usually happens that growth stops as food runs out and waste builds up. So growth rate equals death rate, population plateaus. So the what is exactly happening is at the stationary phase is at stationary phase what happens, the nutrients which is required for the process, for the entire process of the microbial growth, it's usually runs out, and the waste material it builds up, it gets accumulated in this phase. So that's why the growth rate stops. That's why this is known as stationary phase. Then comes the death phase. Death phase is where cell death exceeds growth and population declines. So death phase is where the microbes begin to die because the condition are no longer good. This the main the very certain reason for the death phase and why microbes start dying at this phase is because the conditions are no longer good and longer appropriate for a particular microbes to grow or adjust or divide at this phase. Moving on to the next slide, lag phase, cellular adaptation and preparation. Now, what is usually happening at lag phase? Let's see it again. So, lag phase, no apparent increase in cell numbers, intense metabolic activity and enzyme synthesis. Duration varies from minute to hours. Cell increase in size and metabolic activity. Duration depends on the previous growth condition, inoculum size, environmental factors. Also, in simpler words, what happens at lag phase, the microbes are not growing yet. As I said, it just the microbe just starts adapting to the new environment. It's their initial phase. For example, a new person shifts to a new house, and he that particular person, he or she gradually starts adapting to their phase. So at these so this is these phase so that phase is usually known as their lag phase. So over here also, when we are talking about in microbes, a certain microbes is put into a lag phase where it starts growing and adapting to the environment, just like any human being start adapting to any new environment when they shift from one particular place to any new place or any new country, or maybe they sometimes people even travel abroad to enhance their career or something. So and that phase is known as their lag phase. Same applies for the microbes. They are getting used to their new environment and preparing to grow. Yes, now moving on to the next slide, exponential phase, maximum growth rate and kinetics. So growth rate equation is equal to micro is equal to in n2 minus in n1 divided by t2 minus t1, where micro is specific growth rate and n is the cell number and t is the time. Generational time is equal to in 2 divided by macro. Time required for population to double, typically 20 minutes to several hours. Optimal condition, maximum growth rate achieved when temperature, pH, nutrients and oxygen are optimal for the species. So over here, what is happening? Let me tell you. So there's, wait a second. So microbial growth kinetics is the study of how fast microbes grow and multiply over time, and what factors affect that speed, such as nutrients, temperature, and waste buildup. So microbial growth kinetics is equally important as microbial growth phases. In order to understand the microbial growth phase, we also need to know the microbial growth kinetics. So microbial growth kinetics is the study of how fast microbes grow and multiply over time, and what factors affect that speed, such as nutrients, temperature, and waste buildup. So the basic concept in simple terms, the growth rates, this tells us how fast the microbes are growing during the log phase. So it's often measured by this equation which is mentioned over here, which is in n2 minus in n1 minus in n1 divided by t2 t1, where n1 and n2 are the number of the cells at time of cells at time t1 and t2. So then there is generational time, the time it takes for the population to double. So generation time means the time it takes for the population to double, which is g is equal to in 2 divided by Micro, then there is exponential growth equation, which is all which is this shows how the number of microbes increase over time in the log phase. So then there is optimal condition. Maximum growth rate is achieved when temperature, pH, nutrients, and oxygen optimal for the species. Yeah. In short, microbial growth kinetics help us predict and control how microbes grow, and how useful it is in food safety, medicine, biotechnology, microbiology. Moving on to the next slide. Stationary phase, equilibrium between growth and death. What is a stationary phase? What is happening at a stationary phase? As I told you that the nutrients run out at this phase and waste builds up. But what else is happening at this phase? Can anybody explain that what else happens at this phase? So limiting factor, nutrient depletion, essential elements consumed, toxic accumulation, metabolic waste products, pH changes, AC to alkaline shifts, oxygen limitations for aerobic organism. Then there is cellular response. Cell may produce secondary metabolites like antibiotics, undergo spoliation or intersurvival modes. This phase is crucial for antibiotic production in industrial microbiology. Yes, it is very much important. Also, at stationary phase where growth stops, the number of the microbes stops increasing. The food is running out and waste is built up. Some microbes start to die, but new ones still grow. So the total numbers stay the same. Now, death phase, population decline and cell mortality. Initial decline, cell death rate exceed reproduction as resource become critically limited. Then there is accelerated death. Toxic accumulation and nutrient starvation cause rapid population decrease. Then there is survival mechanism where some cells from spores or enter dormant states to survive harsh condition. Then the death phase follows first order kinetics, similar to exponential growth, but in reverse. Understanding this phase is essential for sterilization process and antimicrobial effectiveness. Then there is environmental factors. As I said, there is environmental factors that also affect the microbial growth phases. Yes, guys, when certain thing is growing, there is not only one thing which is affecting it, but there are various of environmental factor that affects the microbe when it is in their growth phase. So there is temperature, there is pH, and there is oxygen requirement. So if we talk about temperature, like there is sacrophiles, mesophiles, thermophiles, hyperthermophiles. So these are the temperature at which these bacteria survive. So for mesophiles, it is 20 to 25 degrees Celsius, thermophiles 40 to 80 degrees Celsius, hyperthermophiles 80 degrees Celsius. Then there is pH level. Acidophiles, neutrophiles, alkalinophiles each have their specific pH level. Then there is oxygen requirement, which is obligate aerobes require 0.2, obligate anaerobes killed by 0.2, facultative growth without O2, micro aerophiles low O2 levels. Then there is nutrition factors. Also, if a certain things is going through the growth phase, it also needs nutrition to grow. Not only the factors which affect them, but there are various nutrition which is required for their growth. So even microbes need different kind of nutrition for their growth when they're in their growth phase. They need also there are various conditions in which they grow properly, as well as they need certain nutrition which is essential for their growth, otherwise it won't be able to grow in a proper manner. Same as human beings, if we even don't get proper nutrition in our body, or if we don't get proper food or proper diet, we won't be able to survive, and it won't be beneficial for our, we won't be able to grow in a particular manner. So here is nutrition factors: carbon source, nitrogen and essential minerals. Carbon source, autotrophs, CO2 fixation, heterotrophs, organic compounds like glucose, protein, lipids. Nitrogen source, amino acid, nitrates, nitrites, ammonia or atmosphere, N2 for nitrogen fixing bacteria. Essential minerals, phosphorus, sulfur, potassium, magnesium, iron. Trace elements for enzyme function. So for each particular function, there is a particular nutrition factor. or a particular nutrition, like carbon source, nitrogen source. For carbon sources, why it is for autotrophs, it is for CO2 fixation. Then heterotrophs is for organic compound like glucose, protein, lipids, which is essential for growth. Then there is nitrogen source like amino acid, nitrates, nitrites, ammonia, or atmospheric, which is for nitrogen fixing bacteria. Then there is essential minerals. If there is no essential minerals, no certain microbes will be able to grow properly. So it is very important for a microbe while it's in its growing phase. So phosphorus, sulfur, potassium, magnesium, iron, trace elements for enzyme function. The key takeaway is optimizing conditions for control microbial growth. Now growth monitoring, environmental control. Understanding the 4-phase growth curve enables precise timing for harvesting, sampling, or intervention in microbial processes. Environmental control, temperature, pH, and oxygen optimization can dramatically increase growth rates and yield in biotechnology applications. Nutritional balance, proper carbon, nitrogen, and mineral ratios prevent limiting factors that halt growth in stationary phase. Mastering microbial growth kinetics fundamental to technology, food safety, and clinical microbiology. So, what was the takeaway? The takeaway is mainly growth monitoring. Then there is environmental control. Then there is nutritional balance. So these are the things which is important for a certain microbe to grow when it's in its growth. Any microbe is in its growth phase. A microbe doesn't just simply glow in a closed environment by magic or something special happens inside a closed environment. No, but there's a lot of things which is happening with the microbes, and there are a lot of things required which is essential for the growth of microbe. And as a microbiologist, as a biotechnologist, or even as a science student, we all must know what a microbes go through in its growth phase in order to grow in a closed medium. Also, simple takeaway summary for microbial growth phases and kinetics is microbial growth phase. Microbes grow in 4 phases. As I said, we need to remember this, that it grows in lag phase, log phase, stationary phase, and death phase. Then comes the microbial growth kinetics. What takeaway do we have from microbial growth kinetic is growth rate tells us how fast microbes are growing. Generational time is how long it takes for the population to double. Growth follows a pattern that can be shown with the exponential growth equation. A graph helps visualize these changes clearly over time. Also, let's before ending this video, let's see the graph. This is the microbial growth curve graph. So this is the number of bacteria over here, and this is the time you can see. Then there is lag phase, log phase, stationary phase, and death phase. So this is how you can understand or represent the entire microbial growth phase on a graph. Then there is why it matters. Why are we studying microbial growth phases, and why is it essential, why it matters? Because understanding growth helps in medicine, food safety, biotech, and research. It helps us predict, control, or encourage microbial growth depending on the need. So if you are a microbiologist, or if you are an aspiring microbiologist, or if you are an aspiring biotechnologist, even if you are a simple science student, even if you are a PCB student, knowing this thing is very much essential for all of us, because this thing is beneficial in every certain manner. Because even if you go into medicine, even if you go into food safety, even if you go into research, even if you go into academics, this thing will always help you, and you will always need to know this thing. So this is why it helps us predict, control, or encourage microbial growth depending on the need. So that's all for today, guys. Thank you very much for watching my video. Please like and subscribe my channel. Thank you very much. Stay tuned for the next video. Thank you.